I'm Salemi. Welcome back to Device Talks Tuesdays. Great to have you here. This promises to be a fun, engaging, and informative conversation about the impact of MDR on biocompatibility. Is there a shortage of expertise? We've had a very fun warm-up call, pre-call with this group of panelists, and I know they're eager to answer your questions. So this will be I'll go over the details in a moment, but please get yourself ready for a fun conversation. So first, I'll just do my normal intro. We all know this is a virtual event, but the good news is it's also available on demand on devicetalks.com. So we should have this up there tomorrow, probably afternoon or so. If you've got a colleague or a friend who has missed this presentation but would benefit from it, please have her or him register at devicetalks.com, and they'll be able to watch it in its entirety. They'll be able to ask questions as you will at the, well, as I said, we'll reserve most of them for the end of the conversation, but please fill up our inbox during the presentation, and I'm sure one of these gentlemen will take the question at the appropriate time, and if not, I'll prompt them to do so, and we'll make sure this is an hour well spent for you. The slides are available to you for viewing in the resource widget, so there are some informative slides, some nice imagery from Nelson Labs, so please do check that out as we go along. And if you are tweeting about this, God bless you, thank you. Please use hashtag DeviceTalksTuesday, and it'd be great to get all of the conversation sort of aggregated on the social media. So once again, we are Device Talks. We put on in-person shows someday, most likely next year. But we're very fortunate to have great partners like Nelson Labs to bring their expertise to you wherever you are. And it's been fun to have you all here on our Device Talks Tuesdays program. We do still have our podcast, please. Device Talks Weekly comes out every Friday. You can subscribe on all major podcast channels. We'll have great conversations, continue to have great conversations with leaders in the medtech industry, and chat with our colleagues at Mass Device about news of the week. But once again, thank you to Nelson Labs for putting together this presentation and this great panel. We will be switching from image to image as folks talk and as we sort of walk through this presentation. But once again, please, if there's a question that comes up to you during a slide, feel free to ask it then, and I'll make sure it gets answered. But these are our great panelists here. Thank you to Bo Rollins, Thor Rollins, Matt Jurgensen, and John Iannone for sorry about that, for coming today. I know we talked earlier. We're all at various stages of being vaccinated, and some have felt the effects. Some of us are pushing through it, but we're all here with smiles on our face, and I know it's going to be a really engaging conversation. So with that, I'm going to hand the button over to Thor, who's going to start our very short PowerPoint presentation. Thor, take it away.
Yeah, thanks, Tom. And it will be short, although this might be the only chance that I have to talk without being interrupted, knowing that the other three panelists. So I'm going to take every opportunity that I have, but I will make it short because the whole point of this is to hear from these great individuals we have with us today. So today's kind of presentation or panel discussion really is on the new MDR, and we can expand that to just changes in regulatory scrutiny or way things that are pushed through regulatory bodies, and then the shortage of expertise within the market and how we're seeing that impact us. And I just wanted to give a quick kind of set the base kind of why we're seeing this, kind of let everyone know why we're in this certain area we are right now before we really jump into the questions and discussions. So first off, why now? Like, why are we seeing these issues with the MDR and maybe even with other regulatory bodies or arenas like Asia or FDA? Why are we seeing these shortages? And I get these questions all the time. Can't I just follow the ISO 10993 standard, submit it, and have no questions? Why does biocompatibility have to be so difficult? So, I mean, hopefully after today's discussion, we kind of understand why we're seeing this change and some thoughts on how to approach it. So the reason kind of why, though, is historically, even just a few years ago, biocompatibility was really seen as kind of a checkbox approach. This is the old G95 from the FDA, but the old ISO 10993-1 was very similar, in fact, even more simple. And this chart was the way that people approached biocompatibility. They would just look at the device contact, the contact time, and they just performed the testing that was required. It was really checkbox, and it was really easy. So easy that we would do this for fun as a family. We would go through, and that's how dorky my family is. We would go through fun. We'd pick out a device that we saw on the TV, and we'd go through the charts, and my kids could do it. So it was really easy and simple, and you just submitted those tests, you got results, you submitted it to your regulatory body. And because of that, almost everybody did these types of evaluations in-house, right? So they had people that from QA or regulatory or wherever would do their biocomp, submit the testing, and get approvals. Well, the new standard has come out in 2018, and the new standard is really moving more to a risk-based approach. And there's many reasons that, and all of us on this call today on the panel are part of the ISO, the TC194 working group, which was involved in writing this standard. So there's a lot of reasons why we move this way. It has a lot to do with the types of devices that are coming out, and plus the risk-based approach is just better. The concept really was to try to minimize unnecessary testing on devices that were well categorized, but also understand the testing that was needed for certain devices that are becoming a little bit more complicated that we currently didn't or historically didn't see. So in this new approach, as you can see here, it required a physical and or chemical information gathering exercise. And then based off of that physical and or chemical information, and I do want to highlight the physical part of that first information, you gather that information and then you evaluate what testing is needed. So it was a risk-based approach, helping us looking at the physical and chemical information from your materials and the processes that the device goes under, and that evaluates what testing is needed based on your initial risk assessment. So this is really like people will call it a biological evaluation. Some people call it a biological evaluation plan. Other people just call it a risk assessment, but it's generally the same thing where you take these inputs and then you have somebody who either understands the toxicology or the biocompatibility aspects of 10993 and then write some kind of a risk assessment and plan to mitigate the risk of your medical device. And with this, with these changes, we're starting to see more emphasis, taking emphasis away from some of the old standard animal tests that have been around for decades and putting it more on kind of gathering that information. So like extractable, leachable testing. The problem is that's not really standardized from right now, from lab to lab, and even from regulator to regulator. The FDA is probably going to want something different than notified bodies from Japan. And that will change, I think. It's definitely something we'll talk about as a panel as things become more standardized. But right now, it's not standardized. So the testing you get done right now has to have questions by the regulatory bodies to make sure that they are comfortable with the approach you took. And with an emphasis on that risk-based approach, that requires expertise, not just with your company, but also at the regulatory bodies themselves. And then we'll talk about how cumulative exposure of these devices with the increased complexity increases the difficulties that might go into understanding the risks. And the big impact with MDR, and something that I know both John and Bo have been dealing with a lot, plus, obviously with Matt and I in Nelson Labs, we deal with this too, but specifically with the companies, is we have all these devices that currently were historically been on the market. They've been fine being used. And now we have to evaluate all those devices to the current standards. And what does that mean as far as, testing and just the risk assessments themselves? So this is kind of just a background of why we're here, what kind of things we're seeing. We're kind of moved now. Oh, I also want to point out that this just isn't a notified body thing with MDR. The FDA, and this is right out of their new guidance document, also wants to have a risk-based approach, a biocompatibility evaluation, a plan based on the materials and processes of their medical device. So not just something that we have to think about for notified bodies and MDR, but something that's actually being looked at and enforced throughout the whole world. So with that, we're going to talk a little bit about if we all agree, the four of us, that there is a shortage of expertise, biocompatibility expertise with the MDR. How are we working through the risk assessments with the lack of expertise if we think that is, and really what defines expertise. Like, what is our opinions? And I think a good way to start, let's go ahead and start with Bo, and then we'll just all kind of talk about it. But, Bo, what do you think? Is there a shortage of expertise with MDR? Or do you see that maybe this isn't the case? Do you think there's plenty of people out there who can do these kind of assessments?
Boy, throw me the first loaded question.
Well, I figured that the only chance you get to talk.
Pretty much. No, I think, as far as looking at the volume that right now, especially with MDR going through Europe, the volume of biological evaluations that are being either revised or created, because there are many products that have been in the market for decades that never had to be created to begin with. The amount of work, just volume of work, right? So when you ask about, is there a shortage of expertise? I would definitely say for the volume of biological evaluations that need to be performed, there is a shortage just in manpower of people who are familiar with these concepts, the strategies and the approaches, not taking into mind that when you pull in someone to do this, they have to be familiar with the company itself, right? The manufacturing flow, the processing, suppliers. This is a lot of information for someone to just step in and be able to hit the ground running and figure as part of an evaluation. So we definitely are lacking just the manpower to do this, but also as that leads into the expertise around it. So from my perspective, having to deal with, these timelines of MDR and the volumes of biological evaluations that just need to be revised to evaluate current release of standards and the new gap assessments and things around there, there's just not enough people out there that can come in and have the competency to hit the ground running, to really be able to step in and do a risk-based evaluation and be able to write the justifications and the tie-ins, physiologically speaking, with the use of the devices, because these devices are so diverse. So, I have expertise within my organization and how our devices are used, but if you threw me potentially into a realm, outside of my expertise, I'm not going to know physiologically how those interact and work without doing some baseline. So that's where we're running into this whole shortage. There might be someone out there who's done BEs before, but it doesn't mean that they understand the requirements today and or the use of the device and to do the risks associated with that.
And before we get to the other, and like I said, I just want to add to what Bo said. I know before this 10993, there were lots of people out there that would do risk assessments or biocompatibility, actually, because all they had to do, and they were in the QA regulatory, and they could just follow that box. And I agree. Now it's much more complicated, and it needs a little bit more focus. I don't know, John, do you see the same thing? Do you disagree?
No, I see the same thing. And actually, one of the things that Bo mentioned, I think we really need to highlight is the state of the art, talking about biocompatibility as it is today. Many people have done biological evaluations from whatever the previous guidances were and expectations and based on the previous regulatory landscape, really. But when we talk about MDR, we talk about state of the art, we're really trying to understand the latest and greatest when it comes to those evaluations, what the expectations are, rolling in chemical characterization in a way that most organizations have never thought of or been exposed to, and other organizations that have never even needed to consider chemical characterization. Thor, you pointed out earlier, the process is not a checkbox, right? We're not just doing a test that's on a chart. We're evaluating an endpoint based on data. That alone is a change, right? And where do we get that data? What is that data? Bo was talking about having information about your manufacturing processes, understanding the use of that device, and then pulling that together and gaining that information through, chemical characterization as it's outlined in 1093-18 and so on and so forth. I think that's a big element to identify when we talk about a lack of expertise, because, the bar has been lifted. So that expertise that was there is no longer adequate to meet that bar. So that also started to reduce the number of individuals we can for biological evaluations. One thing I wanted to add, Thor, aside from the expertise that we have available for performing biological evaluations, is there's another element of expertise we have to consider, and that's on the regulator side, right? The expertise there. I know there's a lot going on to make sure that the reviewers are trained, and again, state-of-the-art makes it that much more complicated for everybody on both sides of the table.
Yeah, you led right into my next slide there, John. In fact, Matt, maybe you could take this. Like, what do you think about, and I know you want to be careful, but what do you think about regulatory expertise and what we've seen recently? Not just, obviously FDA, notified bodies, but Japanese Ministry of Health and Welfare, you got the Chinese FDA. Like, what are you seeing right now when it comes to these submissions and the regulatory bodies?
Well, I think that there's a few things that we can say there, and I certainly want to be respectful to everybody. Before we move on too far, I wanted to just kind of touch on one thing in the previous slide where you have a bullet point and it says what defines expertise, and I just wanted to say that's something that we know how we handle that at Nielsen Labs. We send our CVs with our biological evaluations and so on and so forth. But is there a consensus on what needs to be in that CV in order for that person to be counted as an expert to a notified body?
Yeah, that's a great. In fact, I'd love to hear Bo and Johnson. So, obviously the standard says you have to have a combination of education and experience to be able to make these decisions. So it's pretty vague, right? And so, I've been doing Biocomp for 20 years. I know John's been doing it for 20 years. Bo's not even 20 years old yet. So I know that some of us have been doing it longer than others, but also education plays a big role in that. So, Bo, how do you guys do that? Like with your company, when you do your own BEs, how do you guys represent that you're qualified to do so?
No, it's a very good point. So one of the interesting things about MDR that helped with me on this is going back and reviewing BEs that have been done throughout the years. And I'm talking about BEs that were all done to -1 2009. So we're talking about the same standard and seeing the flavors of biological evaluations that have come out of that. So we have definitions and explanations on how to do it, but the actual application within the umbrella of a same organization can be dramatically different. And it's interesting to see that, basically there was a lack of guidance in the standard to begin with, to also define, how this should be done. And that led into basically my approach, and working forward was to set up a structure where we restart the slate. So part of it was revising the SOPs internally to ensure that we're compliant with latest and greatest, right? By doing so, you've erased your slate of authors. And then essentially at that point, you start training. And it's good. How do you prove competency? Well, generally speaking, it's through examination, right? Like you can go to as many conferences and discussions as you want. Doesn't mean that you actually understood or retained the data. So ideally, that's why throughout societies, we determine competency by examination, generally speaking. So that's something that I think on my end, I've been rolling out as an examination for authoring, so that we have a specific criteria where we go through specific trainings to the different parts. And that includes biocompatibility, but also design and risk verifications. So that way they understand the inputs that drive the BE and how it's supposed to run through the whole process. We go through specific trainings on that and how to structure, evaluate physiologically. For our experts, we go through the individual parts and we train to those parts. So that way they have gone through not just read -2 or -5, because we've all read these at some point, but it doesn't mean we understand anything that's in it. And understanding the test, because that's what -1 says, right? You have to be able to review, interpret, and understand the data that comes from these tests. You're never going to be able to do that if you don't actually read and understand. So we go through this process and then we do an examination and then a certification. Maybe that's slight overkill, but at the same time, that's the only way that I think that you can clearly state and show competency is through a robust training and examination process.
It's funny. I'd like to get John's thoughts on this too, but I actually had someone that just said, well, I have a board certified toxicologist. Can I just say that they're qualified? And I can tell you, I have lots of board certified toxicologists on my team, and they're great at evaluating compounds from extractables and leachables. But to try to understand what implantation looks like or sensitization potentially or some of these other endpoints, they wouldn't even know where to start. And so biocomp isn't just about toxicity of a compound. There's a lot more that potentially can go into a biocompatibility evaluation. And so it takes a chemist, it takes a biocomp person, it takes a toxicologist together. And sometimes they can be the same person. But like, for example, Dr. Jorgensen is a PhD in chemistry and he's a board certified toxicologist. So you can kind of hit two birds with one stone. But you need those three disciplines to really kind of have I think, the right type of approach for biocomp. I don't know, John, do you disagree with that?
No, I think that's hitting the nail on the head. Biocompatibility is a cross-functional, multidisciplinary type of expertise. And in order to the original question, going back to that, how to demonstrate competency, I think there's a wide range, and that's by design, by the guidelines. You know, people have established competency again by having a training record on file for the reviewer and having their roles and responsibilities defined, and having perhaps a certificate on file, things of that sort. But I do think that what Bo was pointing out, looking at an examination, right? You pointed out, Thor, a DABT toxicologist. So again, why is it a DABT? Because they went through an examination and they've been certified. So something of that sort helps you to ensure competency of the reviewer. I agree with that entirely.
Yeah. I mean, I don't want to say that has to, I mean, that's the hard part, right? We don't want to create, we're not creating criterias here. That's a good way to do it and have a good confidence that it will be accepted. It's evidence, it's proof, it's self-explanatory. If you have someone who's been doing BEs for 10 years actively, that's their role, their job, that should qualify them as an expertise. It's not like, oh, but they need to go out and take some tests somewhere. It needs to be understood that there's not right now an industry certification, right? We are rolling that out in sterilization. There's the CISS from Amy, so you can get certification in sterilization in specific modalities. Currently, we don't have that for biocomp. So it's not to say that there's a requirement if there's any notified bodies or regulators on here. There's no standardized approach out there that you can go get a certificate. But an organization should be able to determine in their competency of their evaluators internally or subcontractors, because that's another area that no one wants to talk about. You know, with the shortage, we're pulling subcontractors and we're leveraging people that know, historically maybe nothing about your company, the products, the process and that, but they're coming in and writing an evaluation. So they may be an expert in biocompatibility, but it doesn't mean they're necessarily competent or have the expertise to evaluate your type of device in its clinical use. So that it's just something that manufacturers should be aware when they're picking up even subcontractors, to make sure that they've got the experience and the ability to evaluate for the device that's being, evaluated. There's a lot of evaluations in that sentence.
There's a lot of evaluation. That's okay. It's
I
funny.
think it's really important, Bo. I was just going to say very quickly, very important what you pointed out, Bo, is again, there is a wide range of how to meet that requirement of competency. I'm sorry. Go ahead, Thor.
No, I agree. And I was just going to say, I had a conversation actually last week with actually a pretty large company, and they were complaining because their notified body was shooting down their BEs or BEPs because they didn't persuade them enough that they had education and experience to write them. And they're like, we are a large company. We have our name behind us and we're not doing it. So we had to talk through about, in a perfect world, it would be the quality of the document. I mean, if you could write a really good document, that should qualify as one of your experts. But in the reality right now, you have to have some kind of formalized documentation to give out. Like Matt said, we send out our CVs, we send out certificates, and Bo talked about testing. John has his part of it. So there has to be something formalized you're sending out to help the notified body feel confident that this BE or BEP is sufficient. Let's go ahead and move on. Actually, I'll stick with you, Matt, since you pushed us back, but I'm not going to let you off the hook. What do you feel as far as a lack of regulatory expertise right now?
Well, I think that the theme here that we understand is that, biocompatibility has evolved. It's moved from a checkbox approach to one that requires a detailed evaluation. This whole process requires experts on both sides. Certainly with the MDR, we have companies that are producing large volumes of biological evaluations, and that at the same time, regulators, notified bodies need to be able to interpret those. And without, trying to sound negative to anybody, I think there is a shortage of expertise on the notified body side for sure. And the way that we try to approach it, or the way that I think about it, is that, to address that shortage, you need to assume that the person that's reviewing this file is not going to have deep expertise of chemistry and toxicology and a long history of biocompatibility experience. And so how you write that evaluation needs to both teach and lead that reviewer to the conclusion that you want. So it's really to add to the list of things that a good biocompatibility person should be able to do is this persuasive writing, right? You need to be able to frame the problem, explain why risks are mitigated in a way that's convincing to a reviewer, even if that reviewer isn't an expert.
Yeah, John, Bo, do you guys, I agree. Do you guys have any additional inputs there?
The document has to have the rationale. You have to, like Matt said, you have to present what the problem is. You have to explain in the document, why that problem is addressed through the testing or evaluations that you have done, and conclude it. It's not enough to just say, what used to be okay is I have this device, it's categorized this way, I've done these 3 tests or 4 tests or 100 tests or whatever it might be. You really have to explain it from defining a robust, problem statement and identifying through that entire document the rationale and really the mindset through that evaluation.
Yeah. My heart goes out to regulators, to be honest, because if you've ever sat through a tech file review, this individual comes in and they have to go through so many diverse documents and informations and requirements around those. And they're trying to go through and check everything. And yes, it is a bit of a checkbox because they have the base requirements that they have to hit, right, that they have to have there. And who wouldn't have a checklist to make sure that happens? I will also completely agree. What we've seen most success with is when we really get into our evaluations to be standalone and to really tell a story, carry and explain. The days of old where we used to, we cross-referenced everything. You would see, go to this stand, go to this document for this justification, go over here for why this is okay, and see, because of all this stuff, biological evaluation's okay. And it was just a pointing disaster, more or less. Now it's really about going through the intended use, explaining your device, how it's used, educate the reviewer, so that way you can bring them up to a level of understanding and expertise before you get through justifying endpoints and why things are not a risk or are a risk and what you did to mitigate those risks. And I think when you can tell that story, that's where you really get the success around here.
Bo, it actually goes back to what you talked about when you're looking at someone running a BE, which is they have to have some kind of deep knowledge about your device. They can't just jump in and say it's stainless steel and so it's safe, right? You got to know about the processing, you got to know about all these other inputs before you can really get an assessment. So, but these reviewers, you're basically asking them to do the same thing unless you give it to them, right? You're asking them to just jump in without having that deep understanding of your device. And so many customers of ours, they know their device so well that they just kind of think that everybody else knows it that well. And so they'll not give us that background, and they won't give the reviewers that background. So I couldn't agree more. You have to spend a lot of time discussing the background of your device, how it's used, the materials and processes, to really give them enough to feel confident. The only other thing I was going to mention is it does help with a notified body. I think that ties back into our previous slide, which is proving your expertise. If you have someone who writes this that doesn't have a background that they feel confident in, I think we've seen them have more troubles accepting those BEs. But if they feel confident in the person writing them, they tend to be a little bit more accepting of those BEs. So it kind of ties back into education and experience too. So Tom, I think we do have another slide, but I know
yeah, I was going to. We've got two more slides. Why don't you wrap those up? But I did want to let folks know the question queue is filling up. If you have questions, please don't plan on waiting to the end to ask them, because we've got a lot here to work with already. And frankly, you guys are very entertaining, like a well-written Netflix series of the Peaky Blinders of biocompatibility. So I know whatever questions come our way, it's going to be fun to listen to. But again, for our audience, we've got a full house. There are a lot of people out there. If you have a question, ask it now. It'll get answered. If you wait too long, we may run out of time at the end. So do you have a slide or two you want to wrap up with, Thor?
Let's just go. Well, I mean, first off, I'm going to put that on my business card now. But I think let's go ahead and jump to some questions. I mean, obviously, we can go back to this slide if we want to, but I want to make sure people we can talk through some of their questions.
Well, there was a great question about 3D printing from Beth. She asked, how are the regulators addressing biocompatibility using 3D printing technology? Is it similar to injection molded components, or is it more expected? Matt, do you want to take that one first, and then others can follow?
Yeah, I'll just tee off on that a little bit. So, Beth, I think it's a great question. It's one that has a lot of interest to me. So I have a lot of personal interest in 3D printing. The short story is that it really depends on the 3D printing technology that we're talking about. So, for example, 3D printed selected laser sintering of metals has been around for a long time. I don't see a lot of skepticism with these devices. There's a little bit of concern about particulates, but usually these are pretty linear submissions and not a lot of surprises. But as you get towards plastic 3D printed devices, and especially if you are talking about stereolithographic ones where it's photo cured, then there's a high degree of skepticism. The regulators know that these are often very novel materials, so no history or no long history on the market at all with innovative chemistry. There's you need to plan on doing a lot of detailed and careful chemistry and toxicological analysis on those types of devices if they have more than limited contact with the body. Yeah, so it really depends on the type of device, with metal devices being on one end very safe and little skepticism, to photolithographic polymers on the completely opposite end where there's a lot of skepticism, and you ought to plan on giving it some really careful, detailed analysis.
And I'll just add on the photolithographic side of things, sometimes they're still, because they're relatively new companies that with our experience, they're doing those, sometimes have multiple different types of concentrations or mixes or ingredients. And I really think that if you're changing different things like adding colorants to it or whatever, it's like Matt said, it's really important to have a good depth knowledge of chemistry. We talk about even in the chemistry itself, there's a range of risk, I guess you can say, where some devices require more of a material evaluation and maybe some chemistry, and then some devices require really thoughtful and an aggressive approach to chemistry. And I would say that we would want to go more on the higher risk side with those types of devices. John, Bo, do you guys have anything you want to add?
I mean, from my perspective, you guys are hitting right around there. I mean, when we talk about this example, keep in mind it's not also just the material, right? With these inherently, you're looking at, UV, what kind of frequencies, times, and cures if you're utilizing that, or if you have a chemical curing process, whether it's alcohol, dip, sprays. There's a lot of different processes to go through for these to get them to a finished, stable state. So making sure that you're including all those, that's where I think it impacts a lot on the biocompatibility, the stability of these materials. Did you actually reach a cured state? And is it consistent through operations? Because that's going to also run into, and I know that's, process validations, but for Biocomp, we got to look at all of those manufacturing processes, inputs, and flows to see how those impact. So 3D printing can be very straightforward and simple with no additives, very, just laser sinted, put and placed, maybe passivated if it's metal, to being extremely complicated and diverse with multiple additives and processing agents that add variability to it and ultimately can complicate a BE. So I guess I just basically reiterated exactly what Matt said. So
thanks, Bo.
John.
Yeah.
Yeah. The one thing that we can highlight there is really that with 3D printing, especially when we're talking about these more unique type like scaffolds and polymer systems, you have to understand the entire chemistry, not just the ingredients that go in, but what is the end result. That's what we're talking about when we talk about the chemistry there.
Great.
Yeah,
There's a question here that I think just requires one answer from one person, and it was a question I had as well. What is the deadline for MDR compliance? I know it was extended last year. I'm not sure if we reviewed it, but it was going to be next month, right? It's coming up in May?
In May 2021, yeah. And then any device that's expired, their CE marks expired after that, you have to go under the MDR. So. All
right, so that hasn't been moved at all. So it's here, basically.
Unless you guys know something I don't.
I do not know anything. Okay. So let's just walk through. We got a lot of great questions now. I like the phrasing of this one. In the MDR, there isn't a definition of state of art. What is your approach? It just sounds poetic. Who wants to take this one? It just sounds like a real important question to have answered.
I'll start because, and then, I don't know, because I jumped in first, I guess. But this is an interesting, and I know John talks about this a lot, and because it plays an important role in 1093-18, and not just -18, but 1093-1 2018, which is the consensus standard system is kind of broken in Europe. Bo just sent over that -18 was a consensus standard now, right? So there are some work still happening, but the consensus standard system is kind of not working great. And so the standards itself is actually, and I don't think I could ever say this before, but the standards are actually moving quicker than some of the other things. Usually standards take forever, but we're adding things really quickly and changing the standards really quickly. And so for me, it really is, and that state of the art is super important with the MDR because they're not going to follow, at least the ones I've talked to, like TÜV-SÜD and BSI, aren't going to follow the 2009. They're going to follow 2018 because they consider that state of the art. So it is really, for me, more about the standards, which standards are kind of accepted out there, even though they may not be consensus standards. I don't know if you guys disagree with me on that or not, but that's my feeling.
No, I would agree. When it comes to state of the art, a lot of people will think about, the industry best practices, the technology, things of that sort. But state of the art in this case really is understanding the regulatory landscape as it is today, and the most up-to-date standards, the most up-to-date methodologies for doing these evaluations. That really is the state of the art here when it comes to biocompatibility.
I will say that across notified bodies, sorry to just jump in there, guys, but I have seen quite a different definition around the state of the art. There are some notified bodies throughout the EU that have been very adamant about the harmonization list and what's listed there. So if it's not listed on the harmonization, it's not accepted in EU regardless of state of the art. So it has to be accepted before it can be considered state of the art. And then there's others that's like, it doesn't matter where it's from or who publishes, as long as it's a legitimate source, it's state of the art. I think that's a scary end as well, because we're seeing studies and some data coming out of some areas that haven't been, substantiated and or evaluated on more broader, populations or groups. So we could be getting some state of the art requirements coming out that really should not be enforced yet, haven't been properly evaluated, validated or determined, but because they were published, now we're supposed to accept them. It's a slippery slope. I think that, I don't think we need to stay on the other side where we have to wait for a harmonization, because that time frame is so long that we end up really causing impact to our systems trying to meet other regulators. But state of the art can't just encompass anything that's released, anything that's new that comes from some source.
Great. All right, let's move on to a great
I
question.
Agree with you entirely, though. I was just going to add to that real quick. The state of the art, if we think about biological evaluations, they have to really tell that story. You have to have that scientific rationale. If you're leveraging something that's been published and you're able to speak to the scientific validity of it, then that goes back into what Part 1, 1093-1, 2018 speaks to. Whether it's published from whatever source, if you're able to speak to the scientific validity of that information, that's really where you're carrying state of the art. Just because something's out there, I agree with Bo entirely, doesn't mean that that's what you leverage. You have to really tell that story, going back to the biological evaluation. Sorry, Tom.
No worries. Matt, we have a question from Carolyn. What kind of information do you typically see provided for a limited duration device? E&L? Question mark.
I love this question because so for a couple of reasons. So first of all, it reminds us that, in the new 10993-1, we have this row of X's for physical and chemical information, but that applies to all devices with all durations of contact, but that doesn't necessarily mean that every device should have an extractables and or leachables chemistry study, right? So I really want to make that point totally clear, because we see, in our toxicology group, we see a lot of full E&Ls come in for a device that contacts the body for 30 minutes. And they say that they, want a toxicological risk assessment, and there's not really a whole lot that we can do in that context. So full E&L is not for everybody. And if you have a limited contacting device, then there's certainly a good time to pause and say, okay, do I really need this? What do I hope to use with the data, and so on and so forth. But for us, if it's clearly limited and cumulative use couldn't push it into a prolonged category, then we do more of a material evaluation to kind of check that box for chemical and material information, or we'll do a certain type of chemistry testing to try to address CMRs and endocrine disruptors, but not what you would really consider to be a full E&L. And I'm really curious what Bo and John think about this, because I know that you guys see limited contact devices as well.
Yeah,
Did you just go first, John?
We have a lot of questions, so we'll keep them short. Keep the follow-up short. Go ahead, John.
10993-18, which is really the document that helps guide pulling in the chemical information, right, for biological evaluation, speaks right up front about information gathering, as opposed to the actual collection of that data from an empirical source. So Matt, you hit the nail on the head. It doesn't mean every single device gets its own E&L study, full blown. It really means understand the chemical information available about that device, the chemical constituents, the manufacturing process. We sound like a broken record. It all starts from there. And understand, even in certain cases, the physical characteristics of that device and how is that going to impact your evaluation. Either it impacts it from your selection of tests, particular assays. It impacts whether or not you have enough information to speak to an endpoint, so on and so forth. So it's not an E&L study for every device. It's understanding the chemistry. And depending on how you answer those questions, you may need to go further in that collection and possibly with a study.
And I only just want to quickly add that a lot of people need to understand, too, the extraction parameters that are in -18 you typically execute do not really comply and work well for those limited exposures. So if you do have the limited exposures in that, and you're thinking I need to do E&L, you're going to be doing an analysis that is not reflectant of your device's clinical use. So you're going to be getting data that's not appropriate either. It starts you down a rabbit hole that can potentially lead to a lot of issues. So keep in mind, that unfortunately it depends on where you're submitting to and the regulators on what they're going to require around those extraction parameters, but those cause more problems than anything else when it comes with E&L and getting the right situation for your device so that you're getting data that's meaningful to its use.
Or even able to get data, period. Some of those devices you can't even get data from.
So, Thor, we have a great question here from Man. I'm wondering how FDA or NB handle unknowns in chemical characterization. If there are 10 unknowns greater than 1.5 but less than 87 micrograms a day, passing two can you read this for me, Thor?
Yeah, I got it. 2 genotoxicity tests. Yeah. So I actually
don't we just hit delete on these, right?
No, I actually love these unknown questions because in all reality, this is the problem with doing just relying solely on E&L, which is for some devices it's great, but for other devices it can be quite. And it actually goes to your point, Bo, which is let's say you have a limited and I know that changes the AAT, but you have a limited contacting device and you do these really aggressive solvents and you start getting these compounds, and some of them are knowns above the threshold. And now you're stuck dealing with some kind of trying to answer. So the truth of it is that if you do chemistry long enough on devices, you're going to find unknown compounds or tentatively identified compounds that you're going to have to address above a threshold. And there's ways to do that. And you mentioned it in this question. If you look at genotoxicity, you may be able to address the genotoxicity risk, which is, one way to assess that 1.5, like you talked about, because that's a genotoxicity limit. And maybe you can answer some of those questions using a chromosome aberration or a mass lymphoma or an Ames test. But the reality is no one should just take one input into your risk assessment. Chemistry is very important with a lot of devices, but it is just an input into your risk assessment. It isn't the only input into your risk assessment. There should be lots of the material you're gathering, the use of your device, the processing it goes through, and then all the other tests that you're running or evaluations you're running. It tells a story. And so, yes, we have to deal with unknowns, but I find that you have to let the regulatory bodies understand that the chemistry is just an input into your risk assessment and not the risk assessment. I don't know, Matt, you're a board certified toxicologist. Do you disagree with my statement?
No, I mean, the two genotoxicity tests, that comes straight out of ISO 21726. If genotoxicity isn't a risk, then you can use a Kramer class, and that's usually a higher TTC than the 1.5. The other thing I would say is that, if it's coming from a plastic device, then you maybe can look at the rate of release compared to the actual conditions of the study to argue that unknown is actually below a TTC. Or if it's from an all metal device, and you can say, it's a surface residual and its exposure actually isn't prolonged or permanent because it's only on the surface. So there's a couple of ways that you can try to pick at that. But I would say after you try those couple of things, if you have really an unknown above the TTC, you got to go down the path of identifying it. If it's a prolonged or permanent device and you try some tricks, if it's too high, you got to identify it. I've not seen a lot of compromise there.
Matt,
I'll go ahead, John.
I was just going to say very quickly, the challenge of understanding exposure, right, is the first thing that you look at. What is the true exposure to the patient? And the second thing is I'll just go right back to the same thing we've been talking about over and over, that biological evaluation tells a story. And in that story, if you have unknowns, then the question becomes, well, do I understand that this chemistry is too aggressive? Do I have ways to mitigate that risk? Is it biological testing? Is it doing another study that understands better the rate of release? Whatever it is in my toolbox, you have to tell that story, show that scientific progression of solving the problem and coming up with that conclusion. Go ahead, Bill. You wanted to add to that.
I was just going to say, oftentimes your unknowns are not true unknowns, right? Like, yes, the equipment did not identify them. But if you truly go back and look at your process, your product, you're going to find that you can usually identify where those come from, what they should be grouped with, and their degradants, their breakoffs from the extraction process or from other things. So knowing and understanding your process materials things will help you to eliminate the unknowns by grouping them, not eliminating by deleting, but by grouping them with where they come from or understanding. And I have run personally into where, we've had devices where the extracting it in alcohol can create glycols that normally would not be there, and in clinical use would never be there, because we're introducing, actions with our extractions. So that's understanding, that, the extraction process creates compounds and things that potentially aren't there to begin with. It is a mystery box of adventure, that we have to go through when we do E&L. But don't just take an unknown as being like, we've got something that we don't know what it is. It's something that's, contaminated our process. There's a good chance that you can figure out where it is or what it is and attribute it to that.
Oh, man. Bo, can I just I just want to add an exclamation mark to what you said, because I really love that point. I think that sometimes manufacturers, and I love labs, right? I mean, we work for a lab here, but sometimes I think there's a little bit too much emphasis or faith that the lab can ID all this stuff in a vacuum, right? When you have context around the manufacturing and formulation of a device, it becomes a lot more clear. So, and it's exactly what John's been saying the whole time. Like, if you consider the formulation and on the upstream processes, those might not be unknowns. They might be something that should be there, but that the lab can't just pull the ID out of a hat.
And this is being recorded, so Bo can actually point out the first time someone says Bo has a great point. So
we're gonna isolate that audio and send it to you, Bo.
Yeah,
so
it's
you'll have
his
that
ringtone for his phone.
for your ringtone.
Yes,
Exactly.
definitely.
Bo, we've got I want to read this question because I never get to say bad boy on a Device Talks Tuesdays. Raquel asked, the word additives are being defined as a bad boy when in reality medical additives can improve the device stabilization. How to circumvent this with NB? ?
I would definitely say start by identifying it not as a bad boy, but a good boy, right? When you start your evaluation and you're going into your materials and your setup, if you're going, hey, look, we have, PVC and we're adding this catalyst and this oxidizer and this stabilizer because we're going to gamma radiate it. And these are the informations behind this, and we're mixing it, and we know all this, and you're identifying it to its advantages and its use. And you might have tox data on the substrate or the material itself that you're mixing in, or you have to evaluate it, if you don't have all that input, you'd have to look at it once it's combined. But I don't, stand up why you're doing it. Hopefully you're adding this for a reason. Identify what that reason is, why it's there, the advantages of it, and then go through why it's not a risk for the intended use. And you should be able to provide data around it. If you're using a stabilizer that's toxic, well, maybe you're using the wrong stabilizer. I'm not saying don't use a stabilizer, but go look for something that is not toxic. That would be my first, it's the same thing with colorants. When you go to use a colorant, don't go pick the cheapest one that's extremely toxic. Go look for one that maybe is a little bit more that's non-toxic and eliminate that risk, on the device to begin with. But stabilizers, from sterilization viewpoint, I love them. We use them all the time to control our oxidations and things from gamma, to be able to make color changes, so things are prettier, not the yellowing. We have different purposes for them, but they're intended. And when they're intended, we should be able to have justifications why they're needed and data around it to support their use. So I don't view them as bad, and I would say you don't view them as bad. Come out straight out with they have a purpose and they're good for their intent.
Yeah, and I just want to argue one thing is everything is bad at a certain level, right? So, and I totally agree with you, Bo, 100%. But I just be prepared.
Too much Thor is a bad thing. Yeah,
it is. It's like too much sugar. So just be prepared though to talk about the amount you're putting in as far as safe, because everything
Correct.
is bad, right? I agree with you though. I just want to make sure you understand, or everyone understands, that there is a point where even the best additives are bad, no matter how much you put in.
You know where this is all coming from? This is all, where this is all coming from is, again, moving that bar, and when it comes to understanding materials, right? They're saying it's not enough to know that something is polyethylene. You have to understand the additives that went into making that device. And I don't think by calling that out and saying evaluate that from a biocompatibility perspective means that it's because it's something bad or that it's something good. They're just saying know the full chemistry. Know the chemistry of that device and understand, like you said, Thor, is it adequate for your particular process, for your particular intended use? Biocompatibility is defined by intended use. So what may be considered biocompatible for one intended use is not going to be biocompatible for another use. Same thing when we talk about the chemistry.
And more cowbell is never a bad thing. So just so we're clear on that. So we're at 5 minutes to 5. We are going to go a little bit beyond 5. We have a lot of questions. We, though, will provide all of these questions that are submitted to our panelists, and they have committed to getting back to you with answers. So please keep the faith. If you have some questions, please still submit them, but we may not get to them in this talk. This is a question, John, I'll pass this one to you. In a risk assessment, I have seen use of different body weights for adults. What to use, 50 kilograms, 70 kilograms?
We actually were talking about this just earlier.
That's why I brought it up. Yeah, it was a heated debate.
Yeah, it depends if you're Bo or if you're me.
All right, point counterpoint, go.
That's right. That's right. I think the important point there is when we're talking about a safety assessment, you need to be conservative. You need to think about the worst case, and you have to be realistic. I think 70 kilograms is not conservative in most cases, and unless you have a justification for using 70 kilograms, then you probably should be more conservative. I think 50 or 60 kilograms is what we were all discussing, actually, is an appropriate body weight to use for adults. And again, it's in the spirit of ensuring safety and being conservative enough.
Yeah, and so I joke because we were arguing about this right before we came on, but the truth of it is the heavier you are, the safer you are from toxic compounds, right? The heavier the body weight you are, so make sure you get all fattened up. But when we write tox assessments, we will, especially if it's used for any population, adult population, there'll be 70 kilograms for adult males and 58 kilograms from adult females, and that's out of 1093-17. Now, if you want to average that out and do 60 kilograms for an adult, I don't think there's anything wrong with that if you have justifications of why you're doing it. I think, like John said, you just want to be conservative. Ultimately, you're the one that's going to have to answer to that level, that limit you put in there. So if your device is only used in overweight Americans, then maybe you can go, 100 kilograms. I don't know. But if you don't have that specific information, then you're going to have something to justify the choices you make.
Good answer. All right. Question for the group. Can we skip the biocompatibility testing if the psychochemical tests do not reveal the presence of any toxic substance?
You have to think about the endpoint. You have to think about the endpoint that you're evaluating and whether or not having chemistry and chemical information will allow you to speak to that particular endpoint. When we think about some of those endpoints, for example, implantation. If we're thinking about implantation and we're understanding a device's potential at the interface between device and patient, if there's going to be some type of a reaction, well, there are other things that can cause that reaction other than just the leaching of a chemical. And this is why one of the reasons why physical chemical information was highlighted in part one, because there are certain physical characteristics to the surface of a device that may actually help to promote certain types of interactions. So again, I'll say, if you have chemical information, fantastic. That's where you should be starting with in many cases. In other situations, you may also need to have physical information in order to proceed with your safety assessment. I think going back to what Bo was saying at the beginning, understanding your device in its entirety and the way it's used, and then everything that is being exposed to the patient, and then understand those endpoints, whether or not you can speak to them from evaluating the chemistry.
Excellent.
100%.
So this may be our last set of questions, but there's three of them, and they all kind of hit upon the same theme. In speaking to the title of the shortage of expertise, one person asked, what training do you recommend to become a biocomp expert? Someone else asked, for aspirants in the field of biocomp, can each of you propose some kind of certified training which can boost carrier aspirations and opportunities? And then someone finally had asked earlier, how do we address this shortage? So maybe we can close out with just sort of some career advice for folks out there who came to learn how to enter this exciting field that you guys are representing so well.
Yeah, Bo, do you want to start? Maybe all of us will just kind of give our opinion.
Yeah, I would say so that was like a couple different things in that question, right? If you're looking to develop yourself, the first part is definitely understanding human physiology, understanding the standards, what the intent of them are and how they work together. You can self-teach yourself very much, a large amount of this content. Going through the ISO standards, they were written to educate and to guide and direct. and then you can continue on to develop yourself. If you're looking for training, is there external trainings? Yes, they're out there. They're typically are very focused on what the standards are requiring, and kind of, a high overview. But if you really want to become an expert in biocompatibility, it has to be self-driven, and you can self-educate through a lot of different facets and through the standards themselves. When it comes to finding places to train and or certify, I'm not, like I said, there's a lot of labs. They tend to be more labs than anyone else that will do trainings and things around competencies. That's better than nothing, definitely. I'm not familiar just right now. There are some consulting firms in that will do trainings and things that will go in depth on site and go through the standards and do trainings for you and do certifications and examinations on those as well. So there are groups that will do that. I don't know if this is really the format to plug anything. So that's what's hard, right? There are, if you find them, if you search for them, you can ask around. There are people who will do them. But ultimately, there's not like an official certification out there or an official degree. And I really liked what, Thor was saying about, like, just because you have a toxicologist, no offense to Matt or other toxicologists, doesn't mean that they understand all the other biological endpoints. Just how I'm not a board certified toxicologist and I'm not going to pretend to know all the toxicology. It takes a family to do these evaluations now. It takes multiple people and inputs, but there's no reason why you can't self-develop yourself in a lot of this area.
John, do you agree?
Yeah, I agree entirely. It's multidisciplinary, right? You have to understand the way everything comes together. Bo, I really appreciated what you said. Understand the standards and then the intent behind those standards. It's all laid out there. The map for development as a biocompatibility expert is there right in the standards. It's understanding the intent of why the chemical characterization is drawn out the way it is, why part one talks about telling that story, why the different endpoints are the way they are, and understanding, not only the tests, like Bo was saying, the actual tests that test labs will train you on, but it's understanding how to gain that information from your own organization, doing that research, looking at the literature, understanding the impact, and self-training. I think, Bo, you're right. The best programs that I've seen are the ones that are developed internal to an organization because they think of it holistically, or the best ones will think of it holistically, I should say. And they're understanding the chemistry, they're understanding the material science, they're understanding the toxicology, and they're saying, how do we bring this all together? It doesn't have to be one person that knows it all. It has to be that here's the team that can handle all of these different elements of biocompatibility.
Yeah, so I actually want to jump on that real fast. The team, if you don't have access to a portion of that team, so like Bo said, if he doesn't have a board-certified toxicologist, there are people out there that you can bring into your team, consultants or hiring or whatever you want to do it, to help with that aspect. But so Nelson, we provide certification training on biocompatibility, but I can tell you and so does other labs, like Bo said, but 3 days of us telling you how these testings are going to do it's not going to be enough. It's just not. You have to kind of, and Bo said self-teach yourself, which I guess is somewhere to say it, but the truth of it, though, is the best way, I think, to learn is actually go with someone and that's been in this industry and try to learn from what they're doing. And I know that's harder to do than say, but the standards are a good way. The intent, like John said, are great. But ultimately, there's so much right now. This is why there's a lack of expertise. There's so much to know and to think about that you either need to kind of have multiple people or someone who's done it for a billion years. But I'm curious. I know Matt's the youngest out of all of us as far as experience go. You've been doing this for 5 years, Matt, and you've been kind of brought into this world of biocomp. What did you find as far as like useful with your training and expertise? Like, what was it for you that was useful?
Yeah, well, I mean, I like everything that was said. And I like Bo, I like to self-learn myself, and I try to do that. But I guess if I were to think about what advice I would give to somebody who was aspiring to get into biocompatibility or aspiring to be a good biocompatibility expert, it would hit on what everybody else has said, that really this is a multidisciplinary field. There's no one person is ever going to be able to do all of it right? And shouldn't really. So if you want to like get into it, you should ask yourself, okay, what really is my core expertise? Where can I truly bring value? For me, I came into this with some strength in chemistry, right? So I knew I could add a little bit to the team when it came to chemistry. We just hired somebody. She's brilliant with blood chemistry, right? And we love bringing that in. There's a person with a niche knowledge in blood chemistry that's just absolutely fantastic. So I would say pick your specialty and look to join a family, and then keep your ears open and learn all the time by listening to what people with expertise outside of yourself say and do. Read a lot of assessments. That's the best advice I could give.
All right, that's great advice. A great way to close out what's been a wildly entertaining Device Talks Tuesday. So thank you, gentlemen, for keeping it lively and fun. I got some nice comments from the audience as well, so I know it was was enjoyed by everyone out there. And this is my opportunity to say thank you to Nelson Labs for putting this together. You're welcome back anytime at this joint. And thanks to everyone in the audience. It's been a great session, a lot of great questions. I do have them here, so take heart, David and Brian and Raquel and Wen. Your questions will be passed on to the experts at Nelson Labs, and perhaps passed on to our other panelists as well, and we'll get you answers for those. So that's it. Thanks everyone for tuning in. Thanks again to the panelists for your time, insights, and entertaining thoughts. And tune in next week. We'll have another great episode of Device Talks Tuesdays for you.